Hello, welcome to today's seminars, which I'm going to discuss lipidomics data analysis. And first I have to see, yeah, this works. A slide about us. So we are Lipotype, we are a service provider using lipidomics technology for quantitative data analysis of lipids, but we also strive to work towards a common goal of lipidomics for a better life. We, of course, analyze lipids, and this is here the full stack of what we do. So we, of course, do lipid analysis, that is, that we get samples, 96-fold format, we extract them, and then robotically infuse them into mass spectrometers. This results into spectra, and these spectra are then interpreted, which is already called data analysis, by an in-house tool called Lipotype Explorer, which then does lipid identification. So it takes the spectra and says, okay, here, this is a lipid, and this has this amount. And afterwards, you can do data reporting, it's called here, but also more analysis. And one of the tools that we have is Lipotype Zoom, but we do all other kinds of custom analysis as well. So what today's webinar is going to be about is not how you get from the spectra to your values, but what happens if you have then an interpreted lipidome, and what do you do now with the data? So what is, of course, what you want is you simply get your lipidome, you do some magic, and you end up with a small number of lipids that completely predict your outcome, and these lipids are also linked to a druggable protein. So dream come true, of course, that's what we all want. And yeah, let's do and start this. What we're gonna do today is we go from data to a result. So, and do the first steps, so there will be nothing fancy today, but just the basic concepts and steps that you can do. And then we look at the basic questions that arise when you do that. So, of course, what you start out with is your data sheet. So that's your tabular spreadsheet of data that you get with samples on the top, and here the features, which are the lipids in this case, lipid species. You might have data values in here, and you might also have just values that are missing in here. Let's first have a look at the first column here. So these are your lipids. And lipids are, these are complex lipids we analyze, and they are a structural heterogeneous group of molecules which are composed of multiple parts. So here you have an example of a glycerophospholipid, and that obviously has then a glycerol as a backbone with a phosphate, and it also has a head group. And these head groups can be many, like cholines or serines or glycerols. And furthermore, you have certain two other positions that can be filled with fatty acids. And these can be from, let's say, 12 to 22 carbon atoms, about from 0 to 6 double bonds. This is one position, and you can have another position that you can have a similar fatty acid, but you could also have not an ester linkage like depicted here, but this oxygen missing, you could have an ether linkage as well. And with all that together, you get a lot of combinations and have a huge number of possible lipid species that you can have. This is a depiction of a plasma lipidome we measured. So out here, you see all the lipid species that we might have measured. Here, these are species and subspecies. In here, you only have the species. These are the lipid classes. And here you have the lipid categories. And lipid categories is, as before mentioned, this is our glycerophospholipids, glycerolipids, sphingolipids, and sterols. But you also have then different classes in these categories. And if you look at one specific, we can here have a little look at the nomenclature, which basically tells you how much you do know about your lipid. So as mentioned before, we have the category, glycerophospholipid, we have a class, this is a choline group, then we have a subclass, which is, we call a PCO or ether-linked PC. And then you enter the realm, what we usually measure with lipidomics. We have a PC with with one ether linkage that has 36 carbon atoms and 5 double bonds. If you go from species to molecular subspecies, you end up with what are the fatty acids, or in this case, a fatty alcohol, 16 carbon atoms, 1 double bond, and one fatty acid, 20 carbon atoms, 4 double bonds. Here you don't know the position, but if you go to the structural subspecies, you actually know this is SN1, this is SN2, so the positions on the glycerol. And if you even go for the isomeric subspecies, you add where the double bonds are. So this is also something you know. Lipidomics usually mostly is that part here. So you go from species to structural subspecies, as highest resolution that you usually get. What we're going to look at today is 4 steps. We do a little bit of data cleaning, data wrangling, then univariate analysis, correlation analysis, and we look also a little bit into enrichment analysis. So this is about data cleaning. The lipotype cat does that. And the idea is, you first have to prepare your data in order to apply whatever algorithms or methods you use later. This is often a lot more than you think, and the bulk of the work that you do, but it's largely unseen later. So you're gonna only see later the fancy method that you use, but not the wrangling, but it might take much more work than you think. So first is that we're gonna have to look at some of the missing values. So as I said before, you have values that are here, but some that are simply missing. And why they are missing is always the question of every statistician. And there could be multiple reasons. One is simply that the lipid might be too low abundant, or simply not be there in that sample. Or the other one is also that they could be missing at random. So there's simply a process or a reason why we don't know. We don't know why we don't know why we don't know. Good. There are different ways of how you can deal with that. So one is you can simply impute these methods so that you come up with a plausible value to put in here. And that could be, you can simply put zeros in there, which is very harsh, or you could find out the nearest neighbors and impute that, or you could, for example, go for the median. But I will argue here that it's actually, you better leave these missing values in here because they basically give us the real picture. But what we should do is that we look at the lipid species that we're having and say, okay, how many missing values do they have? So if you look here, this lipid for this set of samples is only measured once. This doesn't give us much information actually about other samples. So what is done here is that we apply what we call an occupational threshold. Occupation simply means if you have an occupation of 1, this lipid has been measured in every sample. If you have an occupation of 0, this is not measured anywhere. And then you might have it measured once, twice, or every time. And what most people do is they actually look at lipids that are have an occupation of 0.7 to 0.8, so 70 to 80% values, or 20 to 30% missingness, so to speak. This is about the accepted threshold in the field. And that gives you a set of lipids you can work with, and that also will work later with the statistical tests that we do. Another thing you should actually look at is your total lipid amount. So this you simply get when you take all your lipids in one sample and just sum them up. And if you then look at your total lipid amount, you can do a nice histogram like here. And then you just have to look why, how does this look like? And for example, in this case, why do we have some samples over here that are very low abundant and, they have really little lipids in them? And at that point we said, okay, this is plasma. This shouldn't actually be the case. This is probably an error. We need to discard them. On the other side here, as this is plasma, there are people who have really high lipid amounts, which is probably not healthy, but yeah, these people exist, so we should keep them in. That is one way of looking at these total lipid amounts and selecting your samples with. Another thing to think about is simply your replicates. If your replicates now of sample 2, the green one, are all together, this is actually nice. But if, like for the blue one, they're fairly spread out, this will give you quite a large error in your comparison of your error bar, so to speak, and that's probably not giving you good results. Also, if you imagine that you have 2 times exactly the same samples. You have sample 2 and you have another sample 2, which just happen to have the double amount in them. So there might be no difference between sample 1 and sample 2 then, two times the sample, but you have the double amount in them. And that will give you, for every lipid, very significant results, but it's just, this is just not happening. It's because it's, it's still the same sample. So what you need to do is sometimes is to standardize, and best is you standardize before you measure. For example, for plasma, this works very well because you can simply say 1 microliter of plasma is what we look at, and that's then our standard, and that's usually very good to do. But you might standardize by protein if you have that, or you might standardize by number of cells or something else that you can rely on. Let's say for cell culture or tissues or something. What you can always do, you can standardize by molar fraction, what we then call afterwards mole percentage. You simply divide by the total lipid amount again, and then you have the fraction of your lipid from the total. This has its pros and cons, but it's something you can always do, and that will surely help this scenario that I explained here. Good. Another way, what we should do is actually we should get an overview of our lipidome. And one of the overviews that you can take is simply you do a unsupervised dimension reduction, and that can be a principal component analysis or a PCA embedding. That helps you to get these hundreds of dimensions that you have, which is basically every lipid that you measure is called a dimension here, is then reduced to first 1 dimension. And this 1 dimension then represents a certain amount of variance that is represented in your data. And the first principal component gives you the major variance in your data set. So this case is principal component 1. About 30% of all the variance in your data is summarized here. And and then you can go on and do a second principal component and so on. Then if you do a score plot with these principal components, you can have a look, how do your samples perform here? And this is a really beautiful example that basically here we did knockouts of biosynthetic genes. Here's something with with ether lipids, here's something with ceramides, and they just really nicely cluster together and are far apart. And this is what you actually want to see. What this tells you is basically that your experiment is probably successful because the major variation in your data is the experiment you did, in this case, the knockout. But it might also tell you that something is not okay. So for the same data, we had multiple of these knockouts, but had them at different cell densities. We collected them. So these are cells in petri dishes, and we detected some which were sparse and some which were denser. And the first thing we notice in the PCA is that some, that they don't cluster together. They are actually far apart. So the major variance here was rather the cell density, and that we said, okay, we can't work with both of them together. We clearly have to just choose one and separate them individually. The next thing you have is a loadings plot that basically is now all the lipids. Before you had the samples, now you have the lipids, and for every lipid species, the contribution to the principal component is graphed here. And you might see here also if a few things that might help you to interpret if your experiment was principally good. What I like about this is, as I said, this is an ether lipid gene knockout, and what you see over here are all these green circles, while over here there are a lot of green squares. And if you look here, so this is the non-ether lipids, so the standard diacyl PE, and this is the ether link PE. So here you could already see that the major effects are probably what we expect. Good. What you need to do a PCA is, as I said before, in this case, you need to impute your values, because most PCA algorithms don't accept NAs, missing values at all. And this, I would here suggest to do the median, because it's the conservative thing. It basically drags your lipids rather to the middle than spreading them further out over your samples. Sorry. The other thing you need to do is center and scale. That's probably also something the algorithm or the function that you use will ask you, shall I do that? And you say yes, because that homogenizes the lipid species. So basically every lipid species is treated the same way, independent of its amount. If you don't do that, you will have a PCA that is based on only your major lipid species and often this is cholesterol, and often this is about 30%. So we will see nothing but your cholesterol value in here. So it's very important that you look at all lipid species equally and center and scale your data. So as I said, this is what is the dream example. Everything works, everything is nice, but often you just see something like this. So your groups that your experiment is probably based on, blue and red here, are actually largely overlapping, and you don't see much. And also the variance explained is actually here about 25%, so not much in total. So this is very common for plasma data, where the major variation is not your experiment, but the variation between people. But this doesn't mean your result didn't work or your lipidomics analysis didn't work, but it simply means that it's not the major variation in your data set. Good. If we just quickly summarize what we have done here, we looked at data cleaning and wrangling. We basically said we need to handle missing values, we need to check total lipid amount, and we should get an overview. Next, we should be comparing samples, so this or that. And what you need for that, we start out with, is simply two groups. You have lipidomic measurements of either, let's say, control versus treatment, or male versus female. And what you do here is then a statistical test for basically every species that you have. And these tests have assumptions that you should fulfill. And I can tell you already that basically lipid amounts are usually or often not normally distributed. That's what you need for this one assumption for the t-test. What you can do is you can do traitor transformations, but in a way, we usually don't do that, because we think it's more important to look at the lipids as interpretable amounts than just a number that we do analysis with. So what we then refer to, we don't use a t-test, but we use non-parametric tests. And this is either, in this case, the Mann-Whitney test or the Wilcoxon test, which you simply apply. And that works actually quite well if you have enough data. So if you have enough replicates, that means you should have, with, let's say, 10 that works perfectly. And if you have 4, you probably get in trouble. If you have 3, then you can't actually do anything but using a t-test, and have to rely on the robustness of the t-test, which still is actually quite robust outside these normality assumptions. What I would also suggest is that you use paired designs whenever possible. So if you can get away by measuring the same human before and after the intervention, rather than measuring huge amounts of people on one on the other side, that will be beneficial for your test usually. Also, cell culture cells drift over time, and if you can have schemes that work towards that, like paired designs, that helps. So what do you get if you have done a statistical test? Usually this is displayed as a Volcano plot. So here we have human plasma, about 1,000 participants. Here on the x-axis, you have the fold change. That means how much is this, all lipids, how much is every lipid more abundant in females than in males. And here you have the p-value, but this is the negative decadic logarithm of the p-value. So that means the more significant, the lower the p-value, the higher it is on the scale. And this is basically your lipid, your output, but what you usually have is a cutoff. You say, okay, you're actually only interested in things that are significantly different. And you only would look at p-values that are smaller than 0.05. And that gives you another group of lipids you'll be interested in. But this 0.05 means that even if there would be no difference between male and female, you would expect one result in every 20 every 20 tests. So if you do 20 tests, you expect one false positive. But if you do now hundreds of tests, you expect actually quite a few. So what you need to do here is you need to correct for multiple testing. And there might be different algorithms you can use, like very conservative Bonferroni, or you could take the multicollinearity of lipid data into account and use a CDAC test. But most people actually resort to Benjamini-Hochberg, which is simply one way of of looking at this multiple testing correction, which is also called the false discovery rate. And if you look closely, if you go back and back again, there's some values missing here, which is not such a starking example here because we have so many lipid species, we have so many replicates here. But again, this is only a bag of lipids, so it doesn't tell you anything. So you should actually come in and use some lipid knowledge. So what are we actually seeing here? And if we plot them according, let's say, to lipid classes, you suddenly get a different picture. So you would say, in the males, they are all higher in triacylglycerols and deacylglycerols, so their blood lipidomes are much higher in that, which is already a result. Or the females are actually very significantly different in sphingomyelin species. And then you could even drill a little bit deeper and look at these sphingomyelin species and see that all of them, which are highly significant up here, actually have 2 double bonds, which is a signature of a specific sphingoid base in blood. But yeah, so these are results you can get from there. This is obviously an example which works very well because you had 1,000 plasma lipidomes and you can do really nicely p-values up to 30 on the axis. Is what you usually have is more something like this. So we had one of our knockout cell lines that we used, and we have here control, and we have here a sphingomyelin synthase knockout, which performs basically converts a ceramide to a sphingomyelin using PC. And here it's already a little bit more difficult to look at. You might spot some sphingomyelins over here, but yeah, it's more difficult to interpret. So what can you do here? Here we would suggest that you do what you call aggregations or summings. So you simply sum up some lipid species to meaningful chunks. So you could do a class profile. You can do a class profile and standardize to sample. You could look at the total carbon atom profile or the total double bond profile and so on. Or you could even look at the fatty acids and say, what is the fatty acid profile here, and only in the class or in the lipid category and so on. So if we now look at, let's say, the class profile, what we do here is we take all the lipid values that are of the same class and sum them up into one value. And then we can get a class profile. And here you have the classes, and here the sum amount on the y-axis. And if you remember what I told you before, so you have a block here from ceramide to sphingomyelin, and nicely enough, sphingomyelin amounts are strongly reduced. And as there is a backlog towards sphingomyelin, towards the ceramide, there's slightly higher ceramide in that samples, the knockout samples. And as ceramides can't go here, they actually go over here to the hexacyl ceramides, and you actually have more of that. And that's much easier to see in something like a class profile than in a volcano plot. Another example would be the total length profile. And here we have an example where we have an enzyme that only synthesizes ceramides with long fatty acids. And here in your lipid names, you have the total length pointed out. So this is 34, 34, 42. And you see that here the longer ones are reduced and the shorter ones are increased. And yeah, this is also something that is really nicely seen in specifically this summing or aggregation. Yeah, and then you could go and look at others or invent others. So it must be meaningful to your lipidome, basically. So we have rule with univariate analysis. So you need to choose your statistical test. You should examine the volcano plot, account for multiple testing, and explore different aggregation and sums. Next, we look at correlation, and correlation is, so now you don't have two groups, but you have a numerical value, usually from outside, that you try to see if there's anything correlating with your lipidome. So that could be for plasma, it could be BMI, it could be age, it could be a drug that you apply to your cells or whatever. So what you're looking for here is if you have a certain concentration of let's say you have a certain age down here to 400. What does your lipid do on the y-axis, so to speak? And what you do is correlation analysis. There are different methods you can use. If you use a linear method, this is called, usually is the Pearson correlation coefficient that looks if there is a linear relationship between your two variables. If it doesn't have to be linear, it could be also monotonic. That means whenever this increases, this also increases. You have a Spearman rank correlation that you can use. And if you need to use multiple covariables because you are in medicine and you need to account for smoking status, BMI, age, whatever else is important, then you not doing correlations, but you do linear models. And these are not strictly a correlation, but they also give you a very similar result. And they deal with all these covariables that you're having. Good. Here we do one example that is the correlation with cholesterol, and that is an easy target because lipidomes are multi-correlated. They have the feature called multicollinearity that I can't pronounce. But that means that lipidomes are highly correlated within themselves already. And if we now look at the correlation of lipids with cholesterol, and we have here the Pearson's R, so the correlation coefficient, that, by the way, goes from 1 to -1, and 1 is total correlation, basically positive one, that you see that most lipids positively correlate with your cholesterol. And sanity check, cholesterol correlates perfectly with itself, so that is what a perfect correlation would look like. You have an R of 1, p-value of 0, but there are lots of other ones that you could have here, and here we took about 100 plasma lipidomes and did that correlation. So you have lots of sphingomyelins that you could look at. And what you want to look at this, when you look at the scatter plots, you want to see if there's an outlier, for example, up here. And you might ask yourself, should I look at my outliers and maybe do something about them? Or do I have leverage points that are points very out here that simply drive my correlation, but the bulk doesn't do any big influences? So this is nice. The scatter view is nice to actually see if this is okay, if your data are okay. What you usually later have is more of a summary statistic, which is called the forest plot. So you have your estimate of your correlation coefficient and a 95% confidence interval here. And as long as you're far away from 0 with your confidence interval, these should be significant and meaningful. Of course, you can do the same trick as we've had before. You don't have to look only at your species. You can do, again, other correlations. So here with the classes, you see that cholesterol highly correlates with sphingomyelin, but also with PC and cholesterol esters, which is not surprising because there is one very famous reaction in blood plasma that basically takes the fatty acid from PC and puts it on cholesterol to result into a cholesterol ester. Or you look at the fatty acids. That's another way of looking at it. So if you do correlation analysis, you need to choose your type, what you want to look at. You examine your scatter plots, your forest plots, and you again go into correlations of your aggregations and sums and see if you get more general, more summarized, and more meaningful values there. So next is enrichment. And as you see, we have a basketball team at Lipotype that we sponsor and play with, which is actually not the case, but it relates to this example here. So what is enrichment? So enrichment, if you imagine this engineered example, you have a school year and you have students in here that to 1/3 are labeled small, middle-sized, or tall. So there's a blue small one, there's a green middle-sized one, and a red tall one. And if you would form a basketball team from that, you might see that this subgroup of the total school year is probably enriched for red tall students. There's a green one there, but yeah. So this means that this subgroup is enriched for tall students. How do you do this about lipids? There are two ways you do it. You do either feature enrichment, then you tag your lipid species with labels. You say this is a PC class lipid, this is a class sphingomyelin lipid, this one has 18-0 fatty acid, this has a 16-0 fatty acid, and such. You put the labels on there, and later you can see if your subgroup that you're having is enriched for this or that label. If you do pathway enrichment, you need to get labels for pathways, and then you could say, okay, this sphingomyelin has actually a pathway, a label that says sphingomyelin metabolism. And yeah, if you do this, there are different ways of enrichment. So this is simply that you have a subgroup. For this, you need a foreground, which is basically your basketball team, and a background, which is everybody of that school year. And you need certain numbers. So basically, as I said, there is a third of all of your school year were tall people, and here in a basketball team, you need that of these 8 players, you have 7 of them are tall. So these are these K's and N's. And what you do is an open representation analysis based on a Fisher exact test. And here you basically need to know these 4 numbers and plug them into the formula or your algorithm that you're having. And that will tell you, give you a p-value if there is an enrichment or not. If we look again at our example that we had before, we need to choose now a foreground that we do, and we can do this by fold change or by p-value. And in this case, we actually do both. We say this group over here shall be the foreground, so everything that's in control is elevated, and the background is obviously everything. So this what is gray here plus the foreground, and then we plug this in and do a feature enrichment, and yeah, you get surprisingly, you get a sphingomyelin enrichment nicely, because the 6 sphingomyelins that have been in your 500 lipid species all end up in your 60, in a group of 60 over here. And yeah, you get a p-value. This is highly significant. And if you correct for all the tests we've done, yeah, this is still significant. But you also can see that there's a 14-1 fatty acid, which is significantly enriched, or 40 carbon atom length fatty acid in general, but also the LPE class. This is only enriched by significant by p-value, but if you correct, not anymore. But it's still interesting, and you might find this important as a follow-up if this is really something that is working. If you do pathways, as I said, you need these pathway labels, and then you might find that of the 16 sphingolipids that we have in this data set, 6 are labeled over here. These are the 6 sphingomyelins from before, and you would end up with a sphingolipid metabolism enrichment. And if you look this up on KEC, you will get a nice map of your, basically where you use your ceramide and your sphingomyelin, and you could look up the pathways and what happens here. So for the enrichment analysis, we went through what is enrichment analysis and on what it can tell you, basically, 2 kinds. That brings us already to the end of the talk. So we looked at some of the data cleaning, and we looked at univariate correlation and enrichment analysis. But altogether, it's important to know that this statistics is only a tool to an end. What you actually need is to look with your lipidome knowledge, or your lipid knowledge, or the knowledge of your research field into your results and see if there is anything meaningful that statistics gives you back that resonates, that makes sense. Or another big point that you should look at, that lipidomes are always connected. So there's, if you only see 1 or 2 species which are completely different popping out, this is probably not a result you can trust. But if you remember the first result where we had basically all tags increased in male plasma, this is something that really speaks to a lipidome result.